Okay, in this video we are going to be discussing the second part of our protein methods. On the first part we examined the different protocols necessary for purifying proteins and enzymes, and here we are going to be now looking at the methods that we use to visualize and detect proteins. So the first procedure we're going to look at is a method called gel electrophoresis. Now, gel electrophoresis gives us a very powerful method to be able to separate proteins based on their molecular size and be able to stain them so we can actually visualize the proteins in a matrix. So how this is going to work is that we have a particular thin gel substance, which is a matrix. This matrix is made by cross-linking a chemical called acrylamide. What acrylamide does is it forms a plasticized gel that has pores in it. So there is a certain porosity to this gel. These gels are typically about 10 centimeters across and approximately 8 centimeters high. So these are considered mini gels. The mini gels are an invention from the older ones, which were very, very large, which could be anywhere from a foot to 2 feet wide to a foot to 2 feet tall. But with the advent of new chemicals and new separation techniques, the mini gels suffice. Now, the way a gel electrophoresis works is that you have wells at the top of this gel. The gel is poured between 2 glass plates. So the thickness of these gels is often on the order of 1 to 2 millimeters. These are located sandwiched between 2 glass plates, and the wells at the top are where we add our protein samples. So we will have what we are called standard molecular weight markers. We'll explain these in a second. These are going to give us our standard curve to determine the migration distance of a protein. And then our samples can then be added to adjacent wells. Now, once we place these in these wells, the objective is to have the protein move into this porous matrix of a cross-linked acrylamide, and essentially these samples will be percolating through these pores. Under these conditions, what we would have are small proteins will migrate through this gel matrix faster. Large proteins migrate slower. Now, the reason for this is because this is different than the previous protein purification protocol we saw, which was gel filtration or size exclusion chromatography. The holes in these particular chromatography matrix separated so that the small proteins percolated through those beads at a greater rate than the large proteins. The large proteins move between these beads on the chromatographic method faster. So that in chromatography, the large proteins migrated faster. In gel electrophoresis, the pores on these in this matrix are much larger than the pores in the chromatographic method. What this causes is that the small proteins will be able to move through this matrix quicker. The large proteins, as they're entering the pores, essentially get slowed, their migration, because they have to move in and out of the pores, and it's more difficult for the larger proteins to move in and out of those pores. The small proteins can percolate very fast. Now, the question is how do we get these proteins to move through this gel? This is a vertical gel, and so you might think that gravity alone could pull these through this gel. Well, it probably could if given enough time, but we don't have that much time in our experiments, and so what we are going to do is put a charge across the gel. The anode will be at the top, minus charge. The cathode will be at the bottom. By placing a charge across this gel, we create a voltage. And it is this voltage that is going to drive protein migration. Now, there's a consideration on the isoelectric point of the protein. If in our samples we have proteins that are acidic, which means they have a low pI, if we maintain a pH that is close to neutrality here, these proteins will move toward the cathode. Because these proteins are negatively charged, and so they will move toward the positive pole. But what if in our samples we have a basic protein with a large isoelectric point? If we put a voltage across here, these proteins will move toward the anode, the negative pole. The negative pole being at the top, we will not get separation of these proteins. So what we would like to do is have all proteins in the sample move toward the cathode. In order to do that, we have to normalize the charge on our proteins so that in this solution all proteins equal a negative charge. Now how do we do that? This is where we are going to add a detergent. And this detergent is abbreviated SDS, which stands for sodium dodecyl sulfate. Now, if you look at the name of this detergent, you'll see that there's a sodium salt. Dodecyl indicates 12 carbons, and at the end of this 12 carbons is a sulfate. What this detergent does is when we add it to our proteins, the detergent will bind non-covalently, but a very high affinity interaction between this detergent and the proteins. It will unfold. So the first step is it will denature. Denature being unfolding the protein. This will place all proteins in their linear primary structure, removing all secondary and tertiary structure. The second property this will do is because this detergent is negatively charged, it will add a uniform negative charge to all proteins. So as this detergent binds the proteins, even if a protein has a high isoelectric point, being a positively charged protein, this negative charge will overwhelm that, so all proteins will become negative. What these two features of the detergent allow to do is the first one will make all proteins linear. So that the electrophoretic migration through these pores is not dependent on shape of the protein. So for example, if two proteins have two different final structures, tertiary structures, if they are identical in size, but one is globular, the other is rod-shaped. If they're the same size, they technically should migrate through this gel at the same rate. But the geometry of the protein will influence this migration, such that the rod-shaped protein will move slower. Because of its shape, it gets impeded by the pores of this gel. A globular protein is a tighter structure, and so it will be able to migrate faster. So by denaturing and unfolding all of the proteins, we have them all linear, and they have all of the same linearized structure to move, and so structure is not influencing their migration. The second property is a uniform negative charge. This permits positively charged proteins to migrate into the gel. So that positive charged proteins now will be moving according to their molecular size and not dependent on their charge. Now once we add these samples and we add our charge and we have our migration of our proteins, we need to visualize the proteins. And so we will take this gel matrix and we immerse it into a dye. And the typical dyes we use are called Coomassie blue. This dye binds to the proteins in the gel and turns them blue. And so if we place the gel against a white background, we will be able to see blue bands where the protein has migrated to. There's another type of dye that is called silver stain. And this dye here, silver grains will bind to proteins. This increases the sensitivity so that we will be able to detect proteins at much lower amounts, lower concentrations. So the take-home message from this is that we have different types of dyes and stains that can identify and reveal the proteins where they are in the gel, and they have different levels of sensitivity. Now, how do we compare proteins after they have been electrophoresed? Well, if we stain the gel, let's just draw a couple of wells here. What we will find are bands of proteins. These proteins have migrated toward the cathode, away from the anode, dependent on their combined charge and shape. So the faster moving proteins toward the cathode are going to be smaller. Now, before I indicated that in one of our well, we have to have a standard. And these standards are proteins of exact known molecular weight. Again, the smaller proteins will be migrating first. Now, if we were to graph this migration of these proteins, and we graph them molecular weight versus distance migrated, we would see that the faster proteins are at the bottom, they move fastest, and the larger proteins are at the top. What this graph would look like would be like this. So this indicates that the migration of proteins, the distance that they migrate, is a logarithmic relationship to the molecular weight. So in order to properly generate a standard curve that we can use for comparison, what we need to do is just take the log of the molecular weight. And what we will get on this is a straight line. This is now called our standard curve. And this plot comes from proteins that are known molecular weight, and we measure their migration distance. So now if we have a sample in an adjacent well migrating, and that sample has one protein that's located there, all we have to do is measure the migration distance take this migration distance, identify it on our graph, and then take this over to the y-axis, and this will identify the molecular weight of our sample. So the general purpose of electrophoresis serves two functions. One is that we can see how many proteins are in our sample after staining. If we've done an effective job in our purification, at the end we should only have one protein, which would be represented by one stained band. And we can measure its accurate molecular weight based on its migration distance relative to standards. Now the next visualization method also capitalizes on the principles of electrophoresis, but this is called two-dimensional electrophoresis. So in this procedure, we're also going to have a gel that we will be applying proteins to, and then turning on voltage, and so they migrate to the cathode. But first, what we are going to do is further explore the separation of our proteins by using a pH gradient. So the way this works is that we have essentially a gel matrix with different pH. So the center, we can say, is pH 7. The higher end, we could say, would be pH 11. And the lower end, we could say, is pH 3. And the way we control this and generate this pH gradient so that we have pH 3 and it gradually increases in hopefully a linear fashion as we design it properly to pH 11, is that we use chemicals called ampholytes. These chemicals have very defined pH, and they maintain their pH in this gradient, and so we can generate a pH gradient by using different ampholytes. So ampholytes with a low pH will be at one end and high pH at the other. Now what we are going to do is we are going to spot and place our protein in the very center of this pH gradient. We are going to then again apply an electric field. But the electric field in this case, the cathode will be at the low pH, the anode will be at the high pH. So proteins here at pH 7 will generally be charged, and they will move according to their charge to either the positive or negative terminals. So let's say, for example, we have a negatively charged protein. The negatively charged protein will have a low isoelectric point, so at pH 7 it will be negative. So that means if we apply, once we apply the voltage here, this protein will begin to move toward the cathode. Likewise, if we have a positively charged protein, at pH 7, neutral pH, this is going to have a positive charge, and once we apply the voltage, it's going to move toward the negative terminal. Now the question is will this move all the way to the ends? And the answer is no, because once these proteins reach an environment where the pH of the matrix it's moving in is equal to its isoelectric point, what will the charge be on the protein? It will be 0. So let's say the negatively charged protein migrates in this direction and then stops right here. And if there's no further movement toward the cathode, that means that there's no charge on the protein. So then this is identified as the isoelectric point for the protein. Same situation with the positive protein. It is positive at pH 7. It will move toward the anode. And once it reaches a pH where the protein now has a 0 net charge, that will be its isoelectric point. A 0 charge means it will not move in this voltage field. So the first dimension of this separation is separating proteins by their charge. So as you can imagine, if we have a mixture of proteins here in the center, as we apply this voltage gradient, then we will have proteins migrating out and stop at their isoelectric point. Now the second dimension will be the SDS electrophoresis. And in order to do this, what we're going to do is we will have another gel that we have prepared, but instead of having wells, we will have one large well. This whole matrix can now fit into this one large well. So we have proteins distributed across this gel according to their isoelectric points. We place this on top of this gel, and then we apply current. The proteins will migrate into the gel, and in this case, the second dimension, they will be separated by their molecular weight. So under these conditions, what we typically get after we stain the gel to identify the protein location is, instead of bands, we get spots. And these spots correspond to proteins that have been separated in the first dimension by isoelectric point, by charge, and in the second dimension by molecular weight. Now the advantage to this is that we have a much higher resolution of protein separation as compared to just a one-dimensional gel that separates by molecular weight. By having these two dimensions, we have a much greater chance of identifying different proteins in our original sample. Because picture, if we just loaded our original sample on here, we would just have one lane with all of our proteins. But now we've separated them into two dimensions. The next procedure to identify a protein is called an ELISA. An ELISA stands for enzyme linked immunosorbent assay. The principle of this assay is going to be relying on antibodies. Now recall what an antibody is an antibody has a structure such that it is designed to recognize antigen. So we typically draw our antibodies in a Y structure. We have heavy chains and light chains, but the most important part is the ends of our antibody. And this is involved in antibody recognition. Antigen recognition. Now what is the antigen? The antigen can be considered our protein. So if we have an antibody that is specifically recognizing our protein, they will bind on these regions at the ends. These regions are also called the variable regions of an antibody. This is the region that is specific for the protein or antigen we're interested in. So by having a specific detection tool, we can now identify our protein and quantify it in a mixture. So there's two different methods to do an ELISA. One is called an indirect ELISA. And to do this, what we're going to do is we're going to have wells, and we are going to coat the wells with our protein or mixture. Protein mixture. Now the proteins stick to these wells because the wells have been treated to bind protein. The next step is we are going to be adding the antibody that recognizes our protein. And what we will have are these, if our protein is present, we will have the antibody binding the protein. Proteins that are not recognized by the antibody will now, of course, not bind the antibody. How we detect this now is a second antibody, called a secondary antibody. This antibody recognizes the first antibody. So if we have the antibody bound to our protein, this next antibody will come in and recognize the first antibody. Now the second antibody is going to have an enzyme coupled to it. This is a covalent link between a specific enzyme and the antibody. Next we add substrate for this particular antibody, and when that substrate is cleaved into product, we have color. And we can measure this color by using a spectrophotometer. So the way this principle works is that if our protein is present in this original mixture, our antibody will bind. If our antibody binds, the detection antibody will recognize our antibody's presence. That will have an enzyme which will catalyze a substrate into a product and give us color. If our protein is not present, when we add the detection antibody, it will not bind, and it will be washed away. Then we add our secondary antibody, our detection antibody, and it will not bind because the first antibody is not present. We will get no color, and that will indicate to us that the protein of interest was not in this original sample. We can quantify this because the amount of color we get in this reaction is directly proportional to the amount of protein, of our enzyme or protein of interest. Now the final method for ELISA to detect is called a sandwich ELISA. The principle of this ELISA is the same as the indirect ELISA, but it's just a slightly different configuration of antibodies. So in this case, what we are going to do is we are going to bind the antibodies to the bottom of the plate. These antibodies are specific for our protein of interest. Then the next step is we are going to be adding our protein sample. And if our protein is in this sample, then the antibodies will bind the protein. Now, how do we detect this? We add a second antibody that recognizes the protein. So this antibody, if our protein is present, will recognize the protein also. And again, like in our indirect ELISA, this secondary antibody we're adding has enzyme. And once again, as is in the indirect ELISA, we are going to be adding substrate. And if enzyme is present, we will be making product. And this product is color. So in the sandwich ELISA, again, it's just a slightly different configuration of antibodies. We first bind our antibodies that detect our protein of interest to the well. We add our protein sample. If our protein is in this sample, it binds to this antibody. And then we detect this binding by adding another enzyme antibody detection, secondary antibody. We add the substrate, we get color. If our enzyme or protein of interest is not in the sample, we will not have any bound to this antibody, and therefore the secondary antibody will not see anything. It will be washed away, and we will not have any color indicating that our protein was not in that sample. As in the indirect ELISA, the amount of color generated is directly proportional to the amount of protein present. One final detection method to be aware of is identifying amino acids in proteins. Now, we know that amino acids are linked together by peptide bonds to form proteins. These amino acids are specifically encoded by the genes that encode these proteins and give the information to create the protein. Now, in order to study the amino acids in a protein, we can have two different methods at our disposal. The first is called measuring amino acid composition. Amino acid composition is simply a quantification. So we are going to quantify the total number of amino acids in our protein. Now, the way we do this is we have to break up the protein at the peptide bonds to release all of the amino acids in a tube. So we know that these peptide bonds are very, very high energy. They're difficult to break. They're covalent. And so what we are going to do is we are going to add to a tube our protein. It needs to be pure. We bring this to 6-normal hydrochloric acid, and then we boil this overnight. And these conditions are enough to hydrolyze all of the peptide bonds and generate single amino acids from this mixture. Now, typically what we're going to do is apply this to an ion exchange resin. Recall that the two types of ion exchange resins are anion exchange or cation exchange. Let's say, for example, we are going to use cation exchange resin. Recall that this is a negatively charged matrix, so that positive proteins, positively charged proteins, will bind. We will take our amino acid mixture and apply it to this resin, and then we are going to elute with varying pH. So we will have low pH at the very beginning and higher pH at the end of our elution. We can measure the absorption of amino acids using a spectrophotometer to detect the amino acids coming off the column, and moving in this direction will be our elution profile. So we add all of the amino acids onto this, and then we start titrating, increasing pH to start eluting on the column. What we will see is that, for example, aspartic acid, glutamic acid, will obviously bind very, very weakly. We can encourage a little bit of interaction by lowering the pH to neutralize this, but still it's going to be eluted first. As we move into the various PIs, isoelectric points, considering, for example, glycine is going to be higher, it will elute a little bit later. alanine will be similar to glycine. Now down at this end of the spectrum, you can expect arginine. Arginine is positively charged, and so it's going to bind very tightly to the column until you raise the pH sufficiently that you neutralize the amino acid, and it weakens its interaction and elutes off the column. Now, how do we know that these amino acids are exactly what we're looking for? We have a standard curve. We know the elution profile of all amino acids, so the standard is going to be the elution profile of each amino acid. And then all we have to do is compare the peaks we get to that standard. Now recall, with absorption, this is quantitative, and so I should draw this like that. So this means that the area under the curves will dictate how many amino acids there are. So in this example I've drawn here, we could say that the aspartic acid will equal 1, alanine will equal 1, arginine will equal 1, but notice glycine. The area under this curve is exactly twice the area under these curves, so that means glycine has 2. Now, amino acid composition does not give us sequence information. All it tells us is how many amino acids are present in the protein. For sequence analysis, we have to do a little bit more difficult procedure, because now for sequence, we want to know exactly the pattern of amino acids. We want to know the sequential list of amino acids that make up the primary structure of the protein. So for this, we typically use a procedure called Edman degradation. What Edman degradation does is it allows us to specifically label the first amino acid and identify it without disrupting the other amino acids, so we maintain the sequence, the primary sequence. So in general, how we do this is we will take a series of amino acids in a protein or a peptide. Let's just draw some here. And what we want to identify is the N-terminal amino acid. We don't know what this is, so what we're going to do is we are going to add a label. This label will covalently bind to the N-terminal amino acid. So on this procedure, we will next have the label bound to tyrosine and then the rest of the protein. Once this label is attached, we can do a chemical digestion that will disrupt this peptide bond between the first and second amino acids. With the label on the first amino acid, this becomes a very good leaving group. So the conditions to break this peptide bond can be much milder than and will not harm the other peptide bonds. So by this chemical process, we will have this. The labeled tyrosine will now be released from the protein. Then what we do is we use chromatographic procedures to identify this amino acid. Which one is it? In the process, we have released the rest of the protein, which now the next first amino acid with this N-terminus can be coupled to label. Once this is prepared, then we can go in with our chemical treatment, break this peptide bond between the second and third amino acids, releasing this compound, which we use chromatographic procedures to identify amino acid number 2. This was amino acid number 1. And then we keep going through these consecutive rounds of Edman degradation and identification. And eventually we will be able to identify in sequence the amino acids and the order to give the primary structure of the protein.